By the third hour, the parasite had not killed him. That was the problem. That was the only thing I wrote in the margin of my intake log at the start of the third hour, in the flat shorthand of someone who has watched 100 host species die and knows, to within 40 minutes, how the curve runs. The organism integrates. The host's homeostatic systems register the load. The cascade begins, glucose destabilizes, thermal regulation slips, fluid pressure climbs until the host's own compensatory mechanisms fail to compensate. It takes, on average, between 6 and 18 hours depending on body mass and metabolic rate. The fastest I have ever recorded was 51 minutes. The slowest was a Verity coastal elk, 22 hours, remarkable enough that we published a supplemental note. The human was at 3 hours and his telemetry was flat. I want to be precise about what I mean by flat. Not declining. Not beginning the early slide. I have learned to recognize the way a physician recognizes the color of a face before the collapse. Flat as in, stable. Flat as in, his core temperature had not moved in 40 minutes, his cortisol had climbed briefly in the first hour and then settled, his glucose had dipped and recovered, his fluid pressures had adjusted by a fraction and then readjusted and then simply stopped moving. His respiratory rate was the same as when we had admitted him to the isolation wing 6 hours before that. He was sitting on the edge of the observation bunk reading a paper manual he had asked us to print in his language, turning pages with the same unhurried rhythm as a person waiting for a transit connection. The organism was integrated. We had confirmed it with 3 independent scans. It was not dormant. It was present, metabolically active, doing what it does in every host body it has ever occupied. And the human's body was simply running. I called Director Hale at the board partition and told her the 3-hour mark had passed without the primary destabilization signature. She asked me to rerun the intake scan. I reran it. The organism was there. The human's vitals were unchanged. She told me to extend the observation window to 6 hours and document any deviation. I documented no deviation. At 4 hours and 12 minutes his glucose dipped, held briefly at a number that in a Kathrani subject would have marked the beginning of terminal cascade, and then rose, not sharply, but steadily, the way a ship that has listed in heavy current corrects without a helmsman's hand. His temperature ticked up by a fraction. His cortisol spiked as a body noticing something unfamiliar, then returned to baseline. Whatever his interior chemistry was doing, it was doing it continuously and without apparent effort. At the 6-hour mark I pulled every prior host viability record in the Compact's archive. 112 species. The fastest termination, 51 minutes. The slowest, 22 hours. The median, 8 hours 40 minutes. The curve was not disputed data, it was the most reliably reproduced finding in the organism's entire research history, across 4 separate research stations and 60 years of documentation. The organism was not a variable. The organism was a constant. What varied was the host. The human was at 6 hours. I brought this to Director Hale in person. She listened without interrupting and then asked, carefully, whether there was a possibility of contamination in the scan, whether the organism I was detecting was a dormant fragment rather than a living integrated specimen. I told her the metabolic activity signature was unambiguous. She asked whether prior exposure might have produced partial immunity. I told her prior exposure produces not immunity but a faster termination, the immune response mounts too aggressively and the cascade accelerates. She asked whether body mass might account for the deviation. I told her his mass was unremarkable for his species and would, if anything, predict a marginally faster termination, not a slower one. She told me to continue observation and to begin logging comparative telemetry, the organism's expression pattern against the human's homeostatic markers, side by side, in real time. I did this. And what I saw, over the following 4 hours, was the thing I have spent considerable time since then trying to find adequate language to describe in a scientific report. The organism, when integrated into a host body, does not simply consume. It reads the host's metabolic gradient, the gap between the host body's optimal set point and its actual current state, and it exploits that gap. It widens it. Every host species has compensatory mechanisms, but those mechanisms operate on a finite reserve and a predictable lag time. The organism widens the gap faster than the host can close it, and eventually the gap becomes a distance the host cannot cross. This is not a metaphor. This is the literal mechanism, documented across all 112 species in the archive. What the comparative telemetry showed me, running live on the display beside the partition glass where the board was now watching in complete silence, was that the human's compensatory mechanisms had no appreciable lag time. When the organism widened the gap by a fraction, the gap closed. Not slowly. Not with the characteristic latency I could measure to 4 decimal places in every archived host. Immediately. Or near enough to immediately that the difference, on the chart, was invisible. His body registered the deviation and corrected it before the organism could widen it further. His temperature, his pressure, his chemical balances, his cellular oxygen exchange, all of it running as a continuous, self-correcting system that the organism was apparently attempting to destabilize and finding, each time, that the floor it had removed had already been replaced. At hour 10, I noted something I had not expected to see in any recording in the organism's history. The organism's expression pattern had changed. In every prior host, the organism's metabolic activity escalates in a characteristic signature, a widening of output that mirrors the widening of the destabilization gap. Escalation, then terminal escalation, then cessation. What the organism's telemetry was showing at hour 10 was not escalation. It was settling. Its output curve had flattened to match the human's. It had stopped widening the gap because the gap was not staying open, and whatever adaptive logic it operated on had registered, apparently, that this host's gradient was not a gradient at all. It was a constant. And the organism, calibrated across millions of years of evolution to exploit the space between a body's stability and its failure, had found a body with no such space, and was, for reasons I could only describe as the organism's equivalent of metabolic resignation, holding still. The human turned another page of his manual. Director Hale asked me, very quietly, through the partition intercom, what the organism was doing. I told her it appeared to be stable. She asked me what stable meant in this context. I told her it meant the organism was no longer destabilizing the host. That the organism's activity curve had reached an equilibrium with the human's homeostatic baseline. That I did not have a category in the Compact's archive for this outcome, because in every prior record the organism's equilibrium point occurred after the host's vital signs had already terminated. The organism always stabilized, but only after the host had stopped. This host had not stopped. She was quiet for a long time. I filed my preliminary findings at the end of hour 12. The organism was integrated and stable. The human's vitals were within normal parameters for his species. There was no evidence of immune response, organ stress, or cascade initiation. I wrote, in the summary line, that the human body appeared to interpret the organism's destabilizing load as simply one more variable within its ordinary homeostatic range, and had managed it accordingly. I did not write the implication. It did not need to be written. The 4 members of the board had been standing at that partition glass for 6 hours. They had read the comparative telemetry themselves. The implication was this: the organism had been our best understanding of a universal mortality mechanism, a thing that ended hosts, reliably, across every biology we had ever tested. What the telemetry had shown us was that the mechanism required a host whose internal stability could be eroded. The human body did not erode. It compensated. It compensated continuously, reflexively, and without apparent limit within the observed window, and the organism, finding no erosion to widen into collapse, had simply come to rest inside it. I opened the intercom to the isolation room. I told the human we were extending his observation window and asked if he needed anything. He looked up from his manual. He thought about it for a moment the way a person thinks about a question whose answer is obvious. I feel fine, he said. I've felt fine the whole time. Is that what you were checking? I told him it was. He nodded and went back to his reading. I closed the intercom. I looked at the telemetry display. I looked at the board. I looked at the 12 hours of comparative data on the screen, 112 destabilization curves from the archive, all of them falling, and one flat line running clean and level straight across the bottom of the chart, the organism's output resting against it like something that had finally found ground. I filed the report. I recommended the compact suspend all further host exposure studies pending a full structural review of the viability framework. What I did not file, and what no one in that room said aloud as we walked out, was the obvious follow-on question: if the organism had found equilibrium inside a human body, and the human body had found no reason to object, what, exactly, did that make the human body to the organism? And what did it make the organism to us?